Okay, lymph and immune system. Do a quick kind of little, we did lymph a little bit at the end of yesterday, but I'll recap it today. So then we'll discern between the non-specific and the specific defenses today. So that's going to be the other main thing, is trying to figure out what's specific and targeted toward a disease or antigen versus non-specific of it's just something that your body's going to do to prevent anything from harming you. So that's really what we've got going there. So as far as the lymphatic system goes in the lymph, you should know, remember the capillary network, you have the arterioles going into a capillary and the venules leaving, and any excessive fluid that leaves out into the tissues are going to get subsequently drained through this sump system, the lymphatic system, that's going to remove excessive amounts of fluid. So those are the lymphatic vessels. And so the schematic that you can see here is the lymphatic vessels really are draining. They don't really run in parallel to the veins, but they are very much like veins. So you know that lymphatic fluid is really similar to plasma in that it's this watery content with small molecules. It's left out like big red blood cells, big white blood cells, things like that. I do want to point out that they are similar to veins. They don't really run next to veins. They're similar to veins. Low pressure, returning fluid back to the heart, except for the lymphatic vessels, just return it to the subclavian. Then we'll go to the heart. And then they have valves to prevent backflow. So those are things that they're have more in common with a vein than they would an artery. You should be clear on the uneven distribution of lymphatic return. So we have on the left, into the left subclavian vein is the thoracic duct. The majority of lymphatic fluid is going to get returned back to the circulation via that. A smaller percent is going to be returned via the right lymphatic duct into the right subclavian vein. So they're just going to come back to the vein and then they mix back with the blood by the time it makes it into the heart. Propulsion is real similar to veins in that you use the muscle pump, moving your muscles to help squeeze and squish it forward. The valves prevent backflow, but also the drop in intrathoracic pressure helps to draw in both venous fluid as well as lymphatic fluid. That helps return the flow as well. This picture, I just happen to like this picture showing this more three-dimensional nature, highlighting that the lymphatic system maybe that sits below the green component, that's sort of taking away any excessive fluid that might have escaped the tissues and kind of made it down versus being in a different realm, so to speak, of the arteries, capillary veins. Okay, so this is where we are today, a lymph node. So we left off with this lymphatic fluid escaping at the level of the tissues. It's coming back to these lymphatic vessels, returning to the subclavian vein via the thoracic duct or the right lymphatic duct. But in between, this fluid is going to go through a series of lymph nodes. We have, I'm missing a slide. There are clusters of lymph nodes in various regions, most notably in our neck. So I don't have a slide on this, I should. There are clusters of lymph nodes in our neck. There are clusters of lymph nodes in our armpits. There are clusters of lymph nodes in our groin, our inguinal region. Those regions may increase in size when you're sick or fighting off an infection near those areas. A lot of times people have like a head cold and they always say their glands are filled, but it's their lymph nodes that are enlarged because a big amount of their white blood cells in their immune system is coming to this area that's hosting the infection in essence to try to fight it. So they're going to be enlarged because they're accommodating a lot more cells and fluid to do that. So we have lymph nodes everywhere. It's not just in those regions. Those regions just have to be a high concentration, a larger number of them kind of clustered together. But I want you to visualize this lymph node here and see that you have fluid coming in and then going out the other side. So it's these little nodes. I always like to think of it like car washes. You know, you're kind of in one side, the fluid's gonna go in one side and then out the other like a car would. So we're going to have a middle part where most of the fluid is and then an outside part. The function of the lymph node is we're inspecting lymph for antigens. This is a little bit different. I changed the wording here that was in, I think I said cleansing the blood, but it's not cleaning it. It's really just identifying antigens. So if any fluid escaped at the tissue level, before that fluid is allowed to go back to the blood and recirculate, we want to make sure that there's not an antigen that was out in the tissues as well, some sort of bad guy or infection that we're now allowing to go to the blood, and then we just give it a highway system to go throughout our whole body. So the lymph nodes are really to eliminate non-self cells. We have some leukocytes are going to mature here as well. So the two areas of a lymph node that you should be aware of is the medulla, which is going to be the innermost region, and the cortex, which is going to be the outermost region. We're going to double back onto the lymph node when we get through the specific immune system that we're going to do today, and see the relationship and why T cells are located in the medulla and why B cells are going to be located more in the cortex. So the explanation for that will come later. I just wanted to make sure it's in your notes and that you have that connection point here. So this is a histological slide of a lymph node. We see that middle portion here is a lot more sparse. That's like swampland. That's less cellular components, so more fluid is coming through this middle region. So you have the fluid coming through there, and on the outer cortex, it's a lot more densely filled with cells. So there's the cortex and then the medulla. This is how it's broken up. You can see the medulla just sort of loosely defined as this region, and the cortex defined as this region. Notice within the cortex, there are these little circle regions. These circle regions are known as germinal centers. Later, we're going to figure out that those germinal centers house plasma B cells. So you probably want to write that down now so your notes are complete, so that you're going to want to know that later. But the why, we'll come back to it as well. So your B cells are going to be found in there. So you will be responsible for, on an exam, if we see a histology slide of a lymph node or any of these that we're going to go through, but we'll recap that at the end when we're done with the lecture. Here's another image showing a lymph node up there. You can see the medullary area is a little larger, so there's going to be some variations from slide to slide. So we see a little thinner cortex region, but it still has germinal centers nonetheless that are pretty much as a hallmark of the cortex of the medulla. Spleen is the next lymphatic or immune system related organ or tissue bunch. So we're sort of going through like from lymph nodes to spleen of various tissues throughout the body that have an immunological purpose. So the spleen is the next one. We've already heard of one purpose of the spleen. So you get red blood cells, they're made in the bone marrow, they last only 120 days. So they're about 4 months long, and there's no nucleus, they can't have self-repair, so they're really worn out and tattered. You know, you kind of think of like some old beater car with like the bumper dragging behind it, like it gets pretty thrashed. So the spleen is the location that's gonna sort of pick them up and take them out of circulation. I want you to, I'm going to describe how the spleen is. The spleen as an organ is less dense as we see in a cadaver, because the cadavers have no blood. So it's, I like to think of a spleen, studying a spleen on a cadaver is like trying to study grapes by looking at raisins. Like it's really not really how it is. And so a spleen is more like a real flimsy outer capsule that's like, say, a Ziploc bag. And then the inside is this real diffuse fibrous tissue. It's the loose type that we did in 201, so it's the reticular connective tissue. So imagine that connective tissue as like a whole bunch of gauze or cotton balls, just real stringy. You throw that into the Ziploc bag. And if you were to take, you know, a hose, an arterial hose to send blood in, and then you have another little port that's going to drain it off, you just run fluid through. And if something's kind of snagged up, you know, like the old beater car dragging a bumper, it's going to get hung up on the reticular connective tissue. That's one of the mechanisms that the spleen removes these worn out red blood cells. It sort of just ensnares it in this reticular type of connective tissue. And so a reticular, if you recall 201, the reticular refers to like a spider-like, web-like substance. So this is where that kind of comes into play, because it kind of is like that. You get this hung up red blood cell that's snagged on this reticular fibers, and a macrophage comes over and will actually engulf it and ingest it and break it down. You know, so it's gonna like spit out the iron that gets put back in the circulation, heads back to the bone marrow. It's gonna regurgitate out a bunch of the bilirubin, and then it goes to the liver to get dumped out the bile. So it is like the macrophage is like the spider, like it's just gonna go over and then consume it. So that's what's happening in the spleen as one of its jobs. So that's its old job that we learned about last week in terms of the blood. The new job that you're gonna learn about because of today is because it's this capsule that blood is just flowing through with the intention of snagging up old red blood cells, it also is a perfect opportunity for some other white blood cells to sit in clusters and to kind of inspect the flow as it's going by for potential harmful blood-borne antigens. So if there's any antigen or bad guys that's floating through the circulation, the spleen is an ideal location for some of our immune system cells to spot it. The spleen is taken out quite frequently after people have had car accidents or any type of traumatic injury that's going to compress their abdominal cavity because it's so flimsy. It's this little Ziploc bag with just cotton balls in it, so it can rupture and tear really easily. Because its intention is just to run a bunch of blood in it, you can see where hemorrhaging would be an immediate problem with someone with a ruptured spleen. And when you're a surgeon and you're going in, the surgeon's like, I am not going to stitch together tissue paper. I mean, you can see how trying to do that could create more problems, and it's really not going to be very secure. So it's just safer to just take it out entirely. So you think, yeah, you're going to lose some immunological function. You're going to lose a little bit. Our liver and some other areas are going to take up the slack in that. So it still is going to happen, probably not to the magnitude or capacity that you would have if you had the spleen, because that's its job. But it's better to just take it out than try a weird patch job and then have risk of that patient hemorrhaging later. So that's why spleens are pretty frequently taken out. So that's the story with the spleen. Now let's look at it a little more closely. Inside the spleen, we have these two basic regions, which really match the two basic functions of the spleen. You have what's known as the red pulp, and then you have the white pulp. And the spleen is sort of like, to me, the opposite of the pancreas. If you guys remember the pancreas histology slide, it was all purple, and then there was these white spots that was kind of in there that were the islet of Langerhans. Well, the spleen is like all kind of reddish, and then there's these dark spots, because of course, if we have white pulp, it should be dark. It's like, remember, it's opposite. So because we put it in a dye, the same dye that the blood would be in. So it's picking up, it's dyeing all the nuclei. So really the white pulp is really white blood cells clustered in together. But we see them as dark spots on the spleen because the white blood cells picked up all this dye. So really there are these collection areas of white blood cells interspersed throughout the splenic tissue. So think of the whole spleen as being that reticular connective tissue intended to hang up on red blood cells. And then you have these isolated islands of what's known as white pulp, but we see it as really the dark spot. So it's kind of opposite to what it's named. But that's because of the dye that's got in there. Here's another picture. This is actually more of an accurate picture. This one here has them in such stark contrast, and rarely are they actually that starkly contrasted. It really is, you have red pulp area, and then all of a sudden you have this higher cluster. I always think it looks like someone like sprinkled extra pepper in one area, because really that's what it is. It ultimately is lots and lots of tiny little individual cells bunched together. And so those cells are still out here, but they're just in a real high concentration right in there in that white pulp area. So the test question that you're gonna get on the lab practical would likely be a picture of a spleen and say, or either a picture in picture form or a question form, but really I'm gonna ask the same thing, whether you see a picture of it or not. Tell me the regions of the spleen. Red pulp, white pulp. What do? Red pulp removes old red blood cells from circulation. White pulp inspects the blood for antigens. So that's the basic function of the spleen. The thymus is our next immunological tissue. So the thymus is located in the center of the chest. It's really large in infants, and it shrinks over time as we age. So older people, it's really more not as functional. It's more, when we open up our cadavers, it just looks like connective tissue. It looks like a fatty blob. It still has some active splenic, or sorry, thymic tissue, but it's just really small, and it's easy when you open it just to think that it's just random connective tissue. So it doesn't really look like much. It's not a real distinct, feature, especially in the cadavers that we get are older. So the thymus, larger in children, shrinks as we age and function. This is where lymphocytes, remember lymphocytes is number 2 on our list of circulating white blood cells. Lymphocytes is the second most numerous cell in the blood for white blood cells, but it is the first most numerous, like we have the most of white blood cells of lymphocytes in the body. There's just a lot more of them out in the tissues. Circulating neutrophils is going to be number 1 because they're our first responders. So the lymphocytes, so again, number 2 that we learned about when we saw the blood the other day. Remember, the lymphocytes look like smaller cells, really large nucleus. The cytoplasm look like a little crescent moon. Those cells are going to be our B cells and our T cells. So B cells is a B lymphocyte. A T cell is a T lymphocyte. We can't discern in our slides which one's a B or T. So for the test, you're just going to see one of those called a lymphocyte. Academically, then I'll have other questions on your exam that's going to be pertaining specifically to the functions of B and T cells. But the thymus is where a generic lymphocyte from the bone marrow goes to the thymus and it becomes a T cell or becomes a T lymphocyte. So this is where it gets sort of its T cell education. The thymus has 2 regions. So if we were to slice a thymus, this purple is what it looks like. So it's got lobules. It kind of looks like a pancreas too, but a purple one. So you've got these little white lines. Those are separating little sections of it. So if we zoom in a little further, we'll see a little higher power, a couple individual lobules. You'll see this one here to the left, kind of a tiny one here towards the bottom, one kind of cut in half on the right, and then there's one there on the top. So you'll see each one of those little lobules have a lighter center, that's the medulla, and a darker purple cortex. How the thymus works is we have our basic lymphocytes that come from the bone marrow. When they arrive into the thymus, they arrive into the medulla. So think of the medulla as the train station. That's where the passengers get off. So our lymphocytes get off into the thymus, and then they go out to the cortex. So the cortex is darker because there's a lot more cells located out in there. So it's a higher concentration of cells, so the dye made it more prominent. And then after these cells reach maturity and are now ready to be T cells, it goes back to the medulla, back into the blood, and now it can circulate throughout the body. So that's what's going on there. Tonsils. They're our next set of immunologically pertinent tissue. Our purpose of our tonsils is to protect our entrance for airway. So we have, if you're breathing in, whether it's through your nose or through your mouth, we want to have these inspection stations in case that there is airborne antigens. So these antigens, if they're airborne, will then hopefully get trapped in the crypts or the grooves of our tonsils, and then our body can figure out what has just come in our body, what are we going to do about it, what kind of cells do we need to mobilize. Even though the rest of the antigens went right on in, it's not like it's stopping them, it's just picking up a few that's on the way in that we can kind of figure out and then make an alert, a systemic alert to say, hey, this is what's coming in, we better start fighting it. It also protects not only our airway, but it's also intended to inspect food coming in. And then we have a secondary component in our GI tract for the food too. So we have a number of these different tonsils. Most people are only aware of the ones when they open their mouth and say, ah, but we have a few others as well. This is a picture showing some of the crypts. It's like a side view of the little crypts in there where we have some of our immune system components within the wall, the yellow in this picture, to then figure out what to do with the antigens. The arrangement of the tonsils from your nasal cavity around through your oral cavity forms an actual ring. So you have your adenoid tonsil at the top, you've got the 2 lateral tubal tonsils and the palatine tonsil, and one at the bottom known as a lingual tonsil. So what it looks like is you end up with this ring. You can see the arrangement. So you don't so much notice the ring on the side view, but you can just see their location. But you can see based on the paired structures, the blue and the orange, that if you were going to take a frontal view of it, you can see how we effectively make a nice ring so that any air or food coming in, it has an inspection view from all sides, essentially. So that's the beauty of the tonsil. So the tonsils fall into the category of MALT, called mucosa-associated lymphoid tissue. So it just means as along our mucosa system, lymphoid has the immunological reference in tissue. So it's kind of just a weird name for it, but it's in this family. So we have MALT in our gut, because it's got mucosa. We have MALT along our vaginal tissue, because that's another opening. So there are things that are helping to protect openings to the world to prevent infection from proliferating. So there's respiratory tract, GI tract, other areas as well with mucous membranes. So the one in the gut, it's also known as GALT, with the gut-associated lymphoid tissue, but GALT is part of the MALT. Though there are lymphatic nodules throughout the entire GI tract, but in the ileum, part of the small intestine, there's large concentrated areas of these lymphatic nodules, and they're known as Peyer's patches. And they're visually very distinct. So if you had a small intestine and you cut it open along the tube and just laid it out, you can see very clearly how it's this very odd, unique, kind of the tissue's completely different. So before we go on, what we've done so far is the lymph. We've identified the fluid that's escaped, the pathway it goes back, and then the lymph nodes along the way. So then in the lymph node, there are these inspection stations of the escaped fluid in the tissue, just like the spleen is an inspection station for the blood. So we have to have these little areas that we want to check out if an antigen comes in. Our tonsils are an inspection station for airborne or foodborne antigens. So you can see we have kind of our entrances and our fluid areas covered with various immunological tissues. Now we'll go on with the nonspecifics. The nonspecific component is to say, all right, an antigen is in. We didn't block it out. What are we going to do about these antigens? And so we have two methods to deal with them. We can do it in the non-specific way, which is on this side of the column, or on the right side, we have the specific. That's again with our B cells and our T cells. So we'll go through the list of the non-specific. And you really want to think of non-specific as, what does your body have that will fight any bad guy or any antigen? The specific is, hey, we have some tools, but one element might be, I'm only coded for chickenpox. You could have measles, mumps, rabies, the flu, and that cell is like, not my problem. And so that's the specific. Once they get coded for a specific antigen, that's all they do. The non-specific, like fever, you could have all kinds of different antigens in your body and your body could respond. So it's just a general response to any antigen. So external barriers, even though I said, oh, if it gets in, external barriers sometimes is included in non-specific and sometimes it's not. I include it because I like to emphasize the importance of our skin. And so if you just don't let something in the first place, you don't deal with it. So we underestimate the power and the importance of our skin. The time that you realize how important skin is in someone's defense and survival is when you consider burn victims. So in 201, when we talked about skin, we did, you should have learned about like the rule of nines. And so the rule of nines is a determination of not how deep the burn is, but how extensive. So is it one arm in the front of your torso? And so there's this estimate that you can do of what is X amount or X percentage of your body that has the skin removed. That's done in an immediate emergency situation because you have to assess immediately their volume loss. They're going to be worried about, you're going to be worried about their hydration. So that rule of nines is going to equate directly over how much fluid replacement that patient is going to need. So that's just their immediate live within the next couple hours response. But their long-term and larger implications is the infection component once you get them to the hospital and stuff. And that's where the burn wards have really a lot of protocols to minimize antigens coming into the patient rooms. They're sort of like the people in bubbles, essentially, because they're so immunologically compromised. So I do keep skin and external barriers here as part of our non-specific category. Not all books do that, but I really want to make sure that you understand the skin is an important part of our immune system before that. And so some of the other tricks up our skin sleeve, shall we say, is just acidity. Even like sweat, earwax, those kind of things have an acidic component to them to prevent bacteria from growing, proliferating in some of these areas. Sweat glands, like I said, and then mucus and cilia. If you're going to breathe in a whole bunch of air that might have antigens in it, hopefully you're going to stick it to mucus. And then the mucus, the cilia, can then migrate it out of your body. Internal barriers, this is more traditional non-specific immune system, is the rest of the list. So we'll go through each one of these. So phagocytosis, you guys remember that's a cell consuming another cell and breaking it down. Phagocytosis might be to break down damaged tissue if you have a cut. So it's just doing that to say your own tissue that's damaged, or in its blundering around and consuming debris, it may consume antigens too. So that's the phagocytosis, it's just consuming other cells. Neutrophils, macrophages, eosinophils are the main ones, but there's a few others that also do that as well. Macrophages and monocytes are the ones I refer to most commonly. The next is inflammation. The response to inflammation, we always hear about how horrible inflammation is. Well, inflammation can be really bad on a chronic level. It does a lot of bad things on a chronic level. Acutely, it's really good. You have a site of damage, we want to mobilize all of our fighters to that area of damage. So inflammation is helping to promote healing by bringing all those factors to that area. So once you have an area that's damaged, you vasodilate, bring all of the good guys into the area, and then you want to wall off, use fibrinogen, wall off this damaged area. Macrophages can go to work and clean up any debris damage. So any of that fluid, it's got to then be pulled out and drained away by the lymph. So now we can send it through the lymph, and now it can go through a series of those lymph nodes like little car washes to inspect for any antigens that might have come in. So maybe you get, maybe you're working out at the barn and you scrape yourself with some barbed wire fence. So you have a site of inflammation. Blood cells going to go there, macrophages are going to eat up the torn up skin. You want to get ready for skin repair. But then the lymphatic drainage may identify there was tetanus on that barbed wire. And so now for the lymph nodes, we can then alert our immune system that some antigens, it wasn't just a damaged area from the barbed wire, but there was some pathogens that entered with it. So that's what's really important about areas of damage having adequate lymphatic drainage. This is a little schematic showing the red site of damage, and then you can see, some of the white blood cells escaping across the endothelium, coming in here, so we're really mobilizing them. So over a longer period of time, inflammation then can cause damage. And chronic inflammation systemically can cause huge cardiovascular problems. Natural killer cells, it is a type of lymphocyte. So this is the only thing that there is a little crossover, but it is a non-specific immune defense. Natural killer cells really just sort of go around our body, and they are looking for your own cells that have turned bad. So natural killer cells, and that could be a cell that a virus got infected into. So viruses are sneaky little things. They go into your cell, so it's like they cloak themselves in in you, so that most of your immune system says, oh, that's just you, you're good. And then the virus, meanwhile, can multiply and, so it wreaks havoc, and that's why it's hard to identify. Natural killer cells are going around looking for these self cells that may be corrupted by a virus or corrupted by cancer. Maybe there was replication that took place, and one of the replication sequence went wrong and made kind of a not appropriate cell. Well, we don't want that cell to then replicate and replicate. So natural killer cells are indeed around to try to eliminate those and create cause apoptosis. Apoptosis is a fancy word for cell death. So it goes around trying to help these erroneously created cells to then die so they don't replicate and create cancer. It's one of the theories of why people increase their cancer risk as they age, because as their immune system surveillance becomes suppressed. Not only do you have increased exposure, longer duration to certain chemicals, but then they also lessen in some of their immunosurveillance at that point. These guys kill by direct contact. So they will identify a cell and then kill by connecting with that. So we can see the tumor cell in this picture, the brown cell, and then the natural killer cell is the big blue one. So it's going to directly go to your own tumor cell. It is getting a little assistance from a killer T cell. So the killer T cell is a T lymphocyte. It's part of our specific immune system. So that's, but we'll just focus on the blue cell connecting with that tumor cell. Fever is our next non-specific defense. If you have an antigen in your body and our B and T cells, our specific immune system is trying to figure out a way to attack. How do we find this antigen? What do we do to the antigen to kill it? How do we eliminate it? You know, it takes a lot of stuff to figure out when you have like a new bad guy entering your body that the B and T cells have to go through a process to figure out. So the intention of fever is to say, well, while we have yet to mount a effective response to kill the antigen, let's do whatever we can to slow down the antigen's replication. So by making our body temperature warmer, we then make it harder for that antigen to replicate and make more of. So we try to slow down their invasion, if you will, by just having fever. Interferon are just chemicals that get released in different conditions. If you have an infected virus cell, like one of your own cells, a virus gets in, it's masked by your own cell membrane, and it's starting to proliferate. It's really the cell sending out interferon going, hey, white blood cells, we're the infected ones. It's sort of a way to alert your body that perhaps it's infected, as well as alerting neighboring cells that there is an infection. So it's produced by the host cell, the one that's currently being corrupted. It's also going to try to slow down any development, impede that virus from replicating. It's going to, increase antigen presentation of antigen. We'll talk about that in a little bit. So it's doing things to help mobilize your immune system to these virus-specific infected cells. Complement, on the other hand, is whatever your body's doing, it makes it better. So complement does what it says. So instead of saying complement like, ooh, that's a good looking antigen over there. No, complement works together with your cells to be more effective. So more of like a synergistic response. So it really just enhances what you're already doing. So here's the interferon slide. It's released by the example I gave is primarily these virus-infected cells to protect neighboring cells, as well as bringing lymphocytes to come and attack the infected cells. So its job is to warn other cells, inhibit cells from viruses. So lymphocytes and macrophages can also release them. But for the purpose of interferon, you want to think of it as a damaged cell warning neighboring cells, trying to attack your immune system to eliminate that site of compromise or corruption from an antigen. And then this is sort of an example showing a virus invaded cell. You can see here's the virus in that little diagram. And so the virus, instead of coming in and trying to get its DNA and get your cell to replicate for it and go to the other one, it's going to be, the host cell is going to make interferons that's going to try to prevent that replication process from taking. Complement is just circulating proteins that's going to enhance the processes that are already going on. Inflammation, phagocytosis, effectiveness in sort of destroying these antigens and dilating vessels, which is inflammation. So the complement system plays a role even in our specific immune system that we have yet to talk about, and then the non-specific that we All right, the specifics. So as we go into specifics, we're back to the slides. We covered this. We covered this as the non-specific. And now the specifics over here of our B lymphocytes and our T lymphocytes, B cells, T cells. They are specific because once they've been connected to and identified as their target, a specific antigen, they're useless for any other antigen. So they get coded for one thing and one thing only. And that's where if you either were sick before or you got a shot and gave yourself an antigen in essence, then you're training certain B and T cells to only fight that. And so exposing yourself over time to various illnesses is one way to build up your immunity, but you're going through the work of being sick, and then you create your memory cells so that later you won't be sick again to that same antigen because now you have specific B and T cells that are coded for that. Or you get yourself vaccinations where you're purposely exposing yourself to antigens that are rendered ineffective in harming you, but still providing your B and T cells an opportunity to figure out how to fight it, kill it, make memory cells. Those antigens that we choose to have for vaccinations are ones that you have a higher likelihood of dying or having some sort of permanent disability as a result of exposure. So those are things that why people get immunized for certain things. The specific defenses. So immunization, as we move forward, is about specifying your B and T cells. So if you've got smallpox, the immunization, you've just prepared your body to be able to respond to it. That's all. So you're pre-sensitizing and pre-directing some of these. So here's a little schematic showing the non-specific, this whole side here, versus these two, the B and T lymphocytes are your specific immune system. So how do we get our B and T cells activated? Naive B cells get made and they're sort of waiting to have some bad guy to attack. So they could have a foreign antigen presented to them and then they get exposed to this foreign antigen and they have to figure out, like in the tonsil, here's an antigen, we've trapped it, and then you can actually bring your naive B and T cells to figure out, well, what do we do with it? This looks like this. Let's make a receptor that fits it. And how do we kill it? Can we like send this chemical? Nope, this chemical doesn't work. Oh, let's make this other chemical. So it has to go through that process. Or a damaged cell releasing some sort of stimuli like the interferon. So these are different ways that they can get activated initially. Once a B and T cell have already been coded for an antigen, say you got your smallpox shot when you were little, or the cold that you had, a flu that you had when you were in kindergarten. Say that comes back. Say you get exposed to smallpox because you had a shot, but you've had a shot. Or you get exposed to the flu from when you were in kindergarten, but it's back again in 2019. So what happens is that antigen makes it in your body, and then you have an antigen presenting cell that's going to alert your immune system. Your macrophage, just there, and then the yellow guy there is an antigen. So it shows up in your body, whether you breathe it in, it's in your tonsil, or it came through a cut, or somehow it's in your body. So we have any of our phagocytic cells, could be a neutrophil, could be an eosinophil, anything. I just have macrophage because I use that one all the time. It's going to go over and consume the antigen. It breaks it up, breaks it apart, and then it becomes now an antigen presenting cell because a portion of that antigen is on display on the cell membrane. Not the whole thing, but just a part of it. So now it's on the cell membrane. If you've never seen this antigen before, it makes no difference. Then your body's like, whatever, you have something weird on your head. But if it's something that has been seen by your body before, say the smallpox and you had smallpox vaccination, or the flu from when you were in kindergarten, you have a T cell that would match that. And that T cell would be seen and binds to, has a little receptor made just for that antigen. And so the antigen presenting cell, although it is a non-specific immune system cell, it is then the trigger to get your T cells and B cells to go, hey, an antigen that you're triggered for is now in the body. So you better start making more of your fighters to eliminate it. And this is how it gets started. So this is what it looks like. You have this dendritic cell. That's another antigen presenting cell. So it could be a macrophage. This one just happens to be a dendritic cell. And it's literally presenting the antigen. You can't see it. And so it's docking onto the yellow cell, which is a T cell. So this is how it is presented. So this big blue cell is a macrophage or any of these phagocytic cells. You have that red dot that looks like a pill. That's the antigen. It gets consumed into the cell, broken up and digested. So you can see it's all these little particles. And then some of the particles are put onto the surface, and then the rest are just excreted out, and then it's just waste in the body. T cells. So now let's go into the specifics of, okay, we now know B and T cells, they're a specific immune system. They get triggered to an antigen or self-damaged cell. Once they have been coded for a specific antigen, when a macrophage or other phagocytic cell presents an antigen to them, they know then to mobilize, and that is now in the body. So now let's look at the B and T cell players. So I've been just talking about them as that they're just one cell, but there's lots of different subsets of them. So the target of T cells is cellular abnormalities, infections. T cells also work by direct cell-to-cell combat. And that's important. It's not written on here, and it should be. So it actually has to be physically touching the antigen to kill it. And I make that distinction because B cells are different. So the T is your hand-to-hand fighting, guys. The types, we have a couple of subtypes. We've got memory T cells. So those ones are going to remember how to make the other ones. We have the three main groups, although there's many others, but we're just going to categorize them down. Suppressor or regulatory. Actually, they're more referred to as Tregs, but they're earlier formerly called suppressor T cells. That is that they prevent you from overreacting to antigens. And it's actually meant to protect you from autoimmune reactions. It's trying to end a response, but sometimes that goes haywire when you are attacking yourself. So it's in these suppressor regulatory cells that also are involved in organ rejection. So they're really looking for self cells gone bad. Well, nothing says gone bad than somebody else's heart cell. That's like a totally different person. So when people have a heart transplant, they have to take medications that's actually going to suppress their T cells because that's what's going to be in charge of organ rejection, things like that. So they're immunocompromised just with that. So that's the job of the suppressors. Then there's the helper. The helper T cells say, hey, we got a bad guy here. B cell, did you hear about them? They're in here. You know, tetanus made it in the body. And so, because they get coded for that disease at the start, but the B cells may not have gotten the memo to start mobilizing. So helper T cells help with the coordination of the attack. And the cytotoxic T cells, those are the killers. Those are the ones that are actually doing the direct cell-to-cell death contact. So this is just another list. T cells, we have the naive ones. Those are the ones that are just made, but they haven't been coded for anything yet. Once they've been sensitized, that's when like identified as a specific antigen, then they become the three we already talked about. So they're sort of like the worker bees and then the memory cells. So later, it's the memory cell that will cause the recreation of a lot of these if it's been a long time since you've been exposed to that antigen. Here's another lymphocyte. It would be a T cell that's actually attacking a cancer cell, another one. So you can see they're reaching out, they kind of morph around. The B cells are different. They don't need to touch the antigen directly. The B cells, they make antibodies. Let's circle this here. They make antibodies. They're going to release chemicals that's going to aid the immune response, kind of like some complement type action. They can act as antigen-presenting cells, but we want to know that the main cells to remember for B cells are going to be plasma cells and memory cells. So it is the plasma cell that makes the antibodies. Let's circle this. So we have in this picture, the purple naive B cell. It gets exposed to this green spiky virus. Once it learns and adapts how to respond, identify, respond to, and attack the virus, it becomes a plasma B cell that then makes the antibody that will be sent to it. So the plasma B cell isn't directly going to the antibody or antigen, the bad guy. It makes the antibodies and it shoots them out over to the antigen. In class, I used to have this really cool Nerf gun, but the Nerf guns, but with like the sticky end. So the idea was if you're an antigen, the antibody would stick to you and mark you like here. So the B cell, because it had to have a receptor. So you would get, at the very least, marked, the antigen would get marked by this antibody, and then a T cell could more easily mobilize and come over and eliminate it, or multiple antigens could get on it and then create a toxic environment. That was kind of it. So the antibodies are pretty little, so that's where the Nerf gun bullets kind of came in as far as that analogy. And so the plasma cell would be the one that makes it. That would be like the Nerf gun itself. So the Nerf gun doesn't have to be near the antigen because it sends out the antibodies, the antibody that's going to be in contact. So the B lymphocytes you need to know is naive, same as T. Memory, it's the bottom, same as T cells. They will have memory cells and the plasma cells. So now we'll go into the antibodies. So for the B cells, they make those immunoglobulins. Remember globulins that were in the blood? That was our second most numerous protein. We had the LDLs, HDLs, and we have the immunoglobulins. So these are the immunoglobulins that are circulating. So they are made by plasma B cells. And I have this little schematic because you're going to have the variation is going to be in the little Y region of the antibody. So that's why I have the picture of like a one B cell with a pink one, another B cell with a blue one, another B cell with a green one. So each B cell, they're going to be coded for a different antigen that would match their antibodies that they're making. So there's 5 different categories of immunoglobulins. They're the IgA, IgE, IgD, IgM. They're shaped like a Y. You have a variable region, that's the one that changes, depends on the antibody. That's going to be the pitchfork side, and the constant region is going to be really like the handle side, if you want to think of it that way. So the action of the antibodies is to bind to antigens. So we have 2 different yellow antigens here in this schematic here. And so you can see that 2 different antibodies are going to be required. So each antibody is only going to target a single antigen. And so it could just neutralize the antigen by giving it some toxic chemicals. Several can clump on, so they can kind of agglutinate and clump them together so that the macrophages can just consume the whole thing. Or it destroys it using complement, or it just marks it for T cells to come and get. So here's the antibodies listed again. So you have all 5, A, D, E, G, M. So these are the things that people get tested for when they're getting tested for allergies and things. And you're going to get in a lot more detail in micro about these. I have IgG in a different color because I wanted your focus to be primarily on that because it's the majority of the antibodies. But you also see IgE. They have an allergy type response. I have this other schematic here. You can see these are plasma B cells that are making the little red antibodies. So they're sort of being sent out. You know, let's think of the plasma B cells, the mothership, you know, and they're sort of sending out these little fighters that go off from it. This is a representation of the different antibodies again. So they all look different. This one, IgM, looks like a snowflake. You can see this one here looks like a barbell. So they look different, but they are going to have a variable region at the end and then have a constant region. One of the important things here is the one that crosses breast milk, so it's IgA. That is one way that a mother can protect her young in the region that she's from. So let's just kind of backtrack to not where we have airplanes and we can go across the world at the moment's notice. We know that there's different diseases and harmful agents all throughout the world in different areas, whether it's malaria in some parts, there might be other conditions that are regionally specific. So a reproducing female in that area has a greater likelihood of being exposed to any of these pathogens and has developed antibodies. So when she has a baby and is nursing the baby, the baby gets her antibodies. And so therefore, when the baby gets exposed to whatever disease is in that area, it immediately has antibodies to fight with before it even had to go through the process of creating them. So nursing babies is a really good way to traditionally have protected the children from the local harmful antigens that are in that area. So we're back to, what's this? It's a lymph node, yep. So I put this lymph node here because now I'm going to tell you the story about the B and T cells in the lymph node. I told you I was going to double back. So this is where I'm double backing. So remember when we had the middle, that's the medulla, and that's where the fluid is coming through. That's where the T cells are. And out here in the cortex is our germinal centers, and that's where our B cells, specifically the plasma B cells. Who can tell me why we have T cells in the medulla versus B cells being out in the periphery in the cortex? I'm going to figure out a reason why that might be. It has to do with how they attack the antigens. Exactly. So you both are right. So we have the fluid coming through the middle. So the T cells need to be in proximity, in contact with the antigen. So it's got to be down in the arena, if you want to think of it as like, this is where the fluid's coming through. The T cells need to be in proximity. The plasma B cells, they're not fighting, but they're going to send out antibodies. And the antibodies can be shot out in there and go in there and help the T cells, because as the antibodies go, if they bump onto an antigen, you know, that they identify, it just makes the T cell that much easier. It's almost like paint gun battle. You know, they basically painted the antigen. So the antibody is going to do its job, but then the T cell can turn around and go, oh, there's an antigen with that antibody on it. I'll just snuff it out. So they actually work together, just doing it in a different way. So the B cells don't have to be in the thick of things because they send antibodies that's going to go in. The T cells have to be right there. Okay, the last thing I'm going to cover here is the response time, and just to cover the types of vaccinations. So we have just 2 more slides. So the response time is first exposure to an antigen can take up to 3 weeks. Like you got to see it, figure out how to find it again, how to kill it, what to do with it. It takes a lot. Sometimes you can do it pretty quick. You got a lot of, about 10 days. Other times it's a little tougher nut to crack and you have to do a little bit more research on it. So the exposure time, it says the first exposure, your response time and your ability to respond and overcome and defeat the antigen could be up to 3 weeks. But you made memory cells. So if you get that antigen again, your response time will be really quick. The first exposure, you could be very slow to respond. Second exposure, you're ready for it. You're going to get it and you're going to respond. So the types of immunity are these. Active, you got exposed. Either were sick or you got, say, the immunization that was a part of the antigen for your body to figure out how to recognize. That's active. You actively went through all of that. Whether you were sick and you got the flu shot, that is active immunity. Passive is getting antibodies that somebody else made. So the 2 examples were mom giving it to her baby through nursing, or you can get serum from somebody else that was sick. So for instance, when Ebola was a big deal in the news a number of years ago, killing lots of people. The people that survived, their blood was incredibly valuable because they pulled their blood out, took their antibodies and would give it to the next person that got sick to help them out to fight it. So their bodies are still trying to fight it and overcome on their own immune system scale, but by getting somebody else's antibodies, help them immensely to survive. So the point I'm trying to make is both of them could be a shot. Some people just think a shot's only the active, but you could get a shot or injected with some serum, but you're getting somebody else's antibodies. So the passive nature is some other body made the antibodies that you're benefiting from at this juncture. The active immunity is you creating your own antibodies and therefore creating memory cells down the road for it. That's the last 2. Yeah, so passive, they're natural. Artificial is the serum, so that's the injection. active antibodies, again, natural that you were sick, or you artificial, you get an active sort of vaccination. So that's sort of the summary. Any questions?